Okay. So genetics. This is by far one of my favorite topics to discuss in biology. I love it. I love it for a couple of reasons. The first of which is we get to talk about Gregor Mendel, who was this crazy monk who lived in Austria. He spent his life working with peas. And he was able, just looking at peas, to describe genetic architecture, not just of peas, but of all of us. Because what we found is that the vast majority of our genetic architecture, whether we're talking about prokaryotes or mammals, is largely the same. The same sorts of phenomenons that determine inheritance in prokaryotes determine inheritance in mammals. And it's amazing the way that he accomplished it. So when we talk about genetics, really what we're talking about is the inheritance of characteristics. Now, it's easiest for us to think about characteristics that we can see, right? So we think about inheritance of eye color and skin color and hair color and how tall we're going to get and how large we're going to become. And that's all true, but our genetic architecture encompasses so much more than that, right? It's all of the proteins that we've been talking about up to this point. All of the stuff associated with the electron transport chain and glycolysis. All of the stuff making up cellular structures like nuclear envelope. All of those things are genetically encoded. And they're all transferred from mom and dad to us. So each unique character is referred to as a trait. And generally speaking, at least historically, the way we thought about traits was a single gene, a single non-random code of our DNA controls that character. What we'll understand a little bit later is that it's not quite that simple. So lots of people who were contemporary with Mendel were thinking about heredity, but they approached it from a very qualitative standpoint. Mendel's novel approach was a quantitative standpoint. He approached it mathematically, which I think is super rad. Think about modeling and, anyways, he's a cool guy. That was his novel approach, and he chose the pea to work with, and that was a really fortuitous selection for a couple of reasons. The first of which is peas occur in a bunch of different varieties. So you can get pea plants, garden pea plants that have, for example, white flowers or purple flowers, green seeds or yellow seeds, and you can get a whole bunch of different varieties. And he non-randomly selected 6 characters associated with these pea plants. And what was awesome was, and incredibly fortuitous, each of those 6 characters acted as though, or was physically located on, a different chromosome. Had they been located closely on the same chromosome, his results would have been very different. But again, as some lucky happenstance, he was able to select 6 characters that at least behaved as though they existed on 6 different chromosomes. Additionally, peas grow really, really fast, and you don't worry about killing pea plants, right? So if you'd used people and you killed people off, that would have been bad. And they grow really, really slowly. So he was able to cultivate peas and able to do lots of experiments very, very quickly. And then finally, the reproductive organs of pea flowers are enclosed tightly within the flower, right? So the pollen-producing stuff, the stuff that accepts the pollen, is completely enclosed within flower petals, which means that he could control who was mating with whom. So I'm going to talk to you a little bit about the way that Mendel approached his experiments. So Mendel took some pea plants, and he identified a single character that he was concerned with over a bunch of different experiments. So one series of experiments he only considered like flower color. And in one experiment he only considered seed color, or seed shape, or pod color, or pod shape, or stature, things like that. And he made sure that the plants that he was working with were, quote, true breeding for a given character. That is, if there was a flower that was purple, or a plant that had purple flowers, it only produced offspring that also had purple flowers. Alternatively, if it was a pea plant that had white flowers, it only had offspring with white flowers. So he ensured that these lineages that he was working with were true breeding for the characters that he was concerned with. Purple only gives rise to purple, and white only gives rise to white. Once he had these true breeding populations, only purple flowers and only white flowers, he crossed them. He took a true breeding purple flower and a true breeding white flower, and he mated them. And he saw what the offspring looked like. So in genetic jargon, we call that the P generation, or the parental generation, where a true breeding flower crosses with another true breeding flower. And that produces what's called the F1 generation, or the first filial generation. What was interesting was true breeding purple and true breeding white, all of the offspring had just a single character. No matter how many times he did it, all of them, if it was flower color, had purple flowers. So they inherited just one single physical trait. So the first filial generation had a single character that was the same as one of the parents. What he then did was he took that first filial generation and he crossed the first filial generation. All those purple flowers that were produced from the true breeding flowers, he crossed them, and what he got was purple flowers and white flowers back. So we got back that second character. That looked something like this. He took a purple flower and he crossed it with the white flower, right? And that's the P generation. So we crossed these two, and he only got purple flowers. And that was the F1 generation. What he then did was he took another individual from the F1 generation that also had purple flowers, and he crossed those two, like this. And what he found was purple and white flowers. And that was in the F2 generation, or the second filial generation. Isn't that odd? We get purple and white, we cross those two, and we only get purple flowers. We take those two purple flowers, or two of those purple flowers from that first filial generation, we cross those, and we get back our purple and white characters. So Mendel was a little bit befuddled. And what he found was that with each of these 6 characteristics, the same exact thing happened. If he was considering P color, for example, they're either green or they're yellow. If he takes true breeding green and true breeding yellow and he crosses them, all of the seeds are yellow. If he takes seeds from those first filial generation, he gets back the green seeds with some yellow seeds. And what he decided was, what that means is one of these characters has to be dominant over the other, right? So in the context of flower color, a purple flower has to be dominant over a white flower. Maybe each offspring gets some character from mom and dad, but the purple one was dominant over the white one. So a dominant character is expressed every time it's present. A recessive character will only be expressed in the absence of a dominant character.
Can you repeat that?
You betcha. A recessive character will only be expressed in the absence of a dominant character.
And that's how they skip generations.
Mm-hmm. And then a dominant character will be expressed every time it's present. So what this experiment began to suggest was each organism has 2 copies of each character or each trait. But we already know that to be true, right? Because we've talked about meiosis already. We know that homologous chromosomes exist, that each individual has 2 copies of each gene. And in the context of Mendel's stuff, one of those is going to be dominant, maybe, and one of those is going to be recessive. And if you have both, only the dominant one is going to be expressed. If you have two recessive, then both of those, or that recessive character, will be expressed. What was really interesting, though, was that he produced this constant 3-to-1 ratio. In the F2 generation, right here, where he got back his purple and his white individuals, there were always 3 purples to 1 white. That was a constant genetic ratio. There's a little bit of variation as a product of sample size, but it was either 3-to-1 or very nearly 3-to-1. Okay. So this is, I'm going to blank this out really quickly because I want to show you a Punnett square. Now, if we were to think about, what Mendel did with true reading individuals, we could diagram that using a Punnett square that I think most of you are familiar with, right? And so if we have an individual that is true reading purple, we know that she has 2 chromosomes, and if she's true reading for purple, what must both of those chromosomes carry? The purple character, right? Or the purple allele. And so a true reading purple, we're going to say purple is capital P, and white is a lowercase p, like that. True reading purple must have 2 purple alleles, right? 2 copies of the purple gene. And what Mendel did was he crossed that with a true reading white individual that had 2 copies of the white allele. Now, a Punnett square is a genetic tool that's super awesome. And it's super awesome because it models a bunch of different things. The first thing that it models is meiosis. So what we're saying is, mom has the potential of producing 2 different gametes with respect to this one genetic character, right? Because she has 2 chromosomes. So what we can say is, into this egg cell, mom might segregate this P, capital P. Alternatively, she might create an egg cell that has this capital P. Dad can do the same thing. He can produce a sperm cell, or a pollen grain, in this case, with this lowercase p, or this lowercase p. And then what we can do is we can model sexual reproduction in a very unromantic sort of way, right? So we could say, well, what if this pollen grain fertilizes that egg cell of mom's? What's the genetic combination going to be? It's going to be a capital P from mom and a lowercase p from dad. It is genetic convention to always put the dominant allele first and the recessive allele second, regardless of who donates it. So what is this offspring going to look like? Big P from mom, little p from dad. And this one?
Big P and a little p.
Oh, sorry.
Right? That's probably the way that I wrote it. So it's a big P from mom, little p from dad. Same thing right here, right? Big P from mom, little p from dad. So each of those individuals carries one of mom's chromosomes and one of dad's chromosomes, which is just sexual reproduction. Each of these individuals has the exact same, what's called genotype. Their genetic makeup is the same. They also have the exact same phenotype. They appear the same, right? And what color is this individual? If purple is dominant and white is recessive, what color is this individual?
Purple.
Purple, because she has a purple allele, right? And that's the dominant character. So this is the F1 generation. Oops. That's the F1 generation. Now, in the second generation, what Mendel did was he took this individual and that individual and he crossed them, right? So that would look something like this. This individual crossed with this individual. We could do another Punnett square And say, okay, so dad could produce a pollen grain that has a capital P in it, right? But dad could also produce a pollen grain that has a lowercase p in it. And mom could produce an egg cell that has a capital P or a lowercase p. You okay with that? Okay. So this individual is going to get a big P from dad and what from mom?
Big P.
A big P as well, right? What color is that individual going to be?
Purple.
How about this individual here?
Purple. Oh, big P, little P.
In color?
Purple.
Purple as well. This individual?
That's a big P.
Big P.
Big P, little p, right? And then this one?
Little P, little P.
So purple, purple, purple,
White.
white. So we have this 3-to-1 ratio, right? That's the 3-to-1 ratio. Now that 3-to-1 ratio is the phenotypic ratio. Phenotypic just means your physical appearance. It's the physical manifestation of genes. So, when you cross two individuals that have a dominant and a recessive allele, you're always going to get a 3-to-1 phenotypic ratio, always and forever. But there's another ratio here, right? There is a difference in genotypes. So we have this individual, and it has 2 dominant alleles. That's called being homozygous dominant. We have 2 individuals that have one of each allele, and that's called being heterozygous.